Good morning. I am Naushin Fatima, and today I'm going to explain about microfluidic and biomems. So let us first get an introduction. It is a revolutionary convergence of microengineering, biology, and electronics for next generation biomedical application. That is the basic definition of biomems and microfluidics. So let us first understand about what is microfluidics. So microfluidics means it is a science and technology of manipulating extremely small fluid volumes, microliters to nanoliters. So microliters means 10 to the power minus 8 and nanoliters means 10 to the power minus 9 volumes through a micro scale channeling to very small scales. We are going to understand about microfluidics. And the core concept defined as it is a control and process of tiny amount of liquid using channels with dimension measured in micrometers, enabling precise biological and chemical operations. So this is the basic understanding of microfluidics. And now let us talk about what is biomems. So the full form of biomems is biomedical microelectromechanical system. That is, it is an integration of biology and microelectromechanical system. And here it defines the integration as biological, electronics, and disciplines. Okay, and as we know, it operates in a scale of micrometer structure, that is 10 to the power minus 8. And here are the features. So the features include minimal sample volume. Since we already told it is a very small amount, it means that it has minimal sample volumes and precision control. Like since it is very small volume, we can value it precisely. And then rapid reaction, since we are using only samples of very small amount, it's easy so we can see the reaction rate and what are the changes in it. And portability, since it is compact and small, it is easy for us to carry from one place to another. And let us now about the essential components. Essential components include microchanneling, micropumps, microvalves, sensors, and mixers. So, what are microchannels? For example, consider like you have a microfluid over here, and you need to transfer the microfluid from here to here. So, in order to transfer it from here to here, you need a pathway. So, that is called as microchannels. So, microchannels are used to carry the liquid from one place to another. Micropumps. So micropumps are used to pump the liquid from one place to another. For example, if the liquid is from lower dimension to a higher dimension, you need to pump it so that it reaches the top. And then microvalves. So valves are used for controlling, that is for on and offing. It is opening and closing so that we can regulate the flow of the liquid. And then sensors. So sensors are used to check whether the liquid is flowing or not, so as to control its speed, its density, etc. And mixers. So mixers combine different fluid for reaction analysis. Like mixers, we know that right. Mixers means different substances together. That is why it is defined as combination of different fluids. And fabrication techniques. There are many fabrication techniques, and major are four. They are photolithography, soft lithography, etching, and micromodeling. So these are the basic things, and let us not learn about in detail, and let us consider about others. And the common materials are silicon, glass, and polymers are major used for these type of applications. And then biomems application. So biomems, as we know, it is a combination of mems and bio. Okay, so they are used in medical diagnosis, for example, in DNA analysis, blood glucose monitoring. For example, we use the um, blood glucose level monitoring, like big machine with a small chip like thing where you insert your blood, which defines, which says about your blood glucose level and instinct. For that purpose, we use biomems. Drug delivery. Drug delivery is when you intake a medicine, the medicine reaches the right path and goes and works on the specific organs, right? So that is why we use drug delivery. And lab on a chip. For example, lab on a chip is an integration of electronics and biomedical engineering, where we design the lab in an electronic chip so that we can get a virtual lab like this, and these are useful for rapid medical analysis. So let us see about the advantages and disadvantages. So as we already told, minimal sample required. It is high sensitivity because we use small amount. We do not have to waste a lot of amount in it. It is easy to use. Portability, we can travel it from one place to another at least because it is very small in size. Fast analysis, since it is very small in size, it is easy for us to analyze it. And cost effective. It is very less cost, and we can we don't even have to invest a large capital amount in order to start it. And then the main challenges in achieving this, the main challenges in the fabrication complexity. So in order to create a fabrication of MEMS, we have to go through a lot of process like etching, lithography, photolithography, many things, modeling, etc. So all these, are a very, very vast, complex methods and the integration difficulty. Even though we create all these, we need to integrate in a proper way. That is why we have integration difficulties, channel blocking. So if we include all substances, all the required components in a chip, we need a way to channel it in a right way from particles, cells, or precipitation of microchannels and developmental cost. In order to do all these, we all require initial cost, right? Like we need a proper amount in order to develop all these things, and these are not cheap. These are quite expensive. That's why we can say developmental cost. If future directions and environment, in future, we can expect a point of care diagnosis. That is when you go to a portable device, or even a medical test. For example, like if you feel like you are sick right now, you want to take a test. You have to go to a lab. You have to give a blood test. You have to wait for the result to come, and this might take for like 8 to 10 hours. Uh, when development of microfluidics and Biomems, we'll just get a device in which we just enter our sample of blood and it will simply, do all the lab testing at once and just give it accurate results. And this is what we call it as on-point diagnosis. Wearable biosensors, like already know we all wearables like smartwatches, which continuously monitor health. That is what we define here. And organ on chip. Like in the recent development, we know that MEMS devices are used to pump heart to function all our organs, right? And these for these purposes, we use organ on chip. And then personalized medicine by knowing, okay, by using all these things, we can personalize like what medicines are required for what diseases, and for based on the user, based on the client, we can personalize them and medicines. So that is what it is, and hence we reach the conclusion of microfluidic systems and values. Thank you.